Hello dear students, my name is Dr. Vaishali Ghai and the name of this channel is Tricks by Chemistry Guru. So this video is for class 9 students, one short video for the bio chapter, the fundamental unit of life. So let us start with the chapter without wasting the time. First thing is, when I talk about this fundamental unit of life, very important thing is cell. We know that different type of organisms, they live in planet and they look different from each other. And we can classify them as bacteria, fungi, plants, animals. But they all are made up of a common thing, that is cell. So in some cases, a single cell performs all the functions. We call them as unicellular organisms. In other cases, we may have different cells. They combine together, they group together, and they perform different functions. So in this chapter, we'll discuss first of all about the history of the cell, the discovery of cell. Then we'll move to unicellular, multicellular organisms, its various types, prokaryotic, eukaryotic. So let's start with the chapter. What is cell? We say that cell is somewhat which is the structural and functional unit of living organisms. Now the thing is, this cell, how come into existence? So the term cell was introduced by an English scientist, Robert Hooke, and he has written this word cell in his book Micrographia. He was working basically in the oak tree, Spanish oak tree, and that cork is the basic substance which he was working, and he was examining a thin slice of cork under his self-made primitive microscope. He found that it consists of, or it resembles the structure of a honeycomb. Honeycomb, you find some hexagonal structure. So he found the same hexagonal structure as honeycomb, which consists of very small compartments. Initially, he was worked on dead cells of plants, and it looks like empty rooms. Later, Anton von Leeuwenhoek described free cells for first time in 1674. Basically, he designed some improved microscope for the first time and he worked on this free cell. Robert Brown, who discovered nucleus in 1831, the most important component of cell is nucleus. That was discovered by Robert Brown. He was working in an orchid root cell and he discovered nucleus over there. So this is about the basic discovery and the history of cell. I hope this is clear. Now let us move to the next point, that is cell theory. So what is cell theory? How this came into existence? When I talk about cell theory, we say that it was presented by these two scientists, Schleiden and Schwann. They gave this theory. And according to this theory, they said that all plants and animals are composed of cells, and cell is the basic unit of life. So they explained, they have given a theory to explain about the cell. They said that each and every plant and animal, they are composed of cells, and cell is the basic unit of life, which was modified later, and nowadays we are having modern cell theory which says that all living beings are composed of these cells, and cell basically is a mass of protoplasm which consists of cell membrane, nucleus, and cell wall in some cases. So basically this cell theory was given by these two scientists. They said that all living cells arise from the pre-existing cells. All living cells, that means they have come from some pre-existing cells. So this is about the cell theory. Now, what are the instruments we generally use for study cell? Because cells are microscopic in nature, so we cannot see these cells through our naked eye, right? So we need to study these cells using some special instruments, and we call these instruments as microscope. What this microscope does, microscope basically provide us some enlarged image of that microscopic object, and we can clearly study the structure of cell. So we have a microscope which is an instrument to study cell. which is basically a common optical instrument as it consists of a combination of lenses. There are different kinds of microscope. One is light microscope, which is of two types. One is simple microscope and other is combined microscope. Like if we talk about amoeba, which is unicellular organism, has irregular shape and it changes its shape continuously on the basis of its function. Like if it has to engulf any food or obtain food in the body, it changes its shape. If it has to digest that food, it changes its shape. Finally, if it has to excrete, again it changes its shape. So amoeba or such type of unicellular organisms do not have specific shape, but other unicellular organisms are having definite shape. Let us move to multicellular. The shape of cells depend upon the position in the body and the specific function which they perform. For example, we have blood cells which are round in shape, nerve cells which are a long elongated tube ganglion along with it, ovum spherical in shape. So different cells on the basis of their function may have different shape. They also vary in size. Like it varies from few microns to in centimeters. Most human cells, basically our body, consists of cell in the range whose diameter is around 8 micrometer, 8.2 micrometer. So they vary in their size also. Now we'll talk about the number. On the basis of number, we can say we have unicellular and multicellular. Many of the multicellular organisms have large body which composed of larger number of cells. So I can say that number relates with increase in size of organism. Larger the number of cells, bigger will be the size of that object. So suppose any human being which weighs around 60 kg. So it has more cells as compared to a human body which has less mass. So less mass means less cells. So this is about the unicellular and multicellular organisms. Now let us move further to the another topic, that is prokaryotic and eukaryotic cells. This type of classification is based on the complexity of cell, how complex the cell is. And we classify cell into two categories, prokaryotic and eukaryotic cell. What are prokaryotic cells? They are the most primitive organisms like bacteria, like blue-green algae, cyanobacteria. What is so specific about them? They lack nuclear membrane. That means they do not have nuclear membrane around their genetic material. And we call them as prokaryote. Prokaryote means, pro means before, and karyon word comes from nucleus. Similarly, the genetic material, they are in direct contact with the cytoplasm and there is no such nuclear membrane that bounds nucleus and doesn't allow the cytoplasm. So nucleus is in direct contact with the cytoplasm. So prokaryote do not contain any cell organelle or membrane bound organelle. Now after this we'll move to the next topic, how the transport of material takes place across cell membrane. So we consider that there are three types of mechanism to show the transport of material. We have passive transport, we have active transport, and we have bulk transport. All these three are basically involved the transport of material. We'll discuss one by one. If I talk about the passive transport, it says that this transport is very slow. That means it allows the particles or material but very slowly. It occurs due to change in the concentration. And it takes place by two ways. One is either osmosis or another is diffusion. So when we talk about diffusion, we say that it is a process of transferring of material from higher concentration to lower concentration. Like suppose if you add some copper crystals, copper sulfate crystals in water, you will find that after some time they get dissolved completely in water. This is nothing but the diffusion. Similarly, diffusion of gases. Where do we have higher concentration, the gases will diffuse towards lower concentration and ultimately will be having uniform concentration throughout. So this is diffusion. Similarly, we have osmosis. When we talk about osmosis, we say that it is a process of the transport of material with the help of a semi-permeable membrane, and it changes from lower concentration to higher concentration. For example, if you take raisin, kishmish, and you place in water, you will find that after some time it swells. That means the water enters into it. So basically what osmosis is, I can say osmosis is a diffusion of solvent through semi-permeable membrane from a solution of lower concentration to higher concentration. Like raisin is placed in water, I can say the raisin is having higher concentration in it as compared to the water in which it is placed. So water which is of low concentration enters into this raisin via semi-permeable membrane. So this is the process of osmosis. Next we have is active transport. When we talk about active transport, we say that this process is comparatively fast and it requires the use of energy in the form of ATPs. ATP is adenosine triphosphate. So basically active transport is a fast process, whereas this passive transport is slow process. Active transport but goes only in one direction. In this case, if we talk about diffusion, it goes in both the directions. It requires energy, it does not use any energy. So basically when we talk about the transport of glucose, transport of amino acids, they pass through plasma membrane by the help of active transport mechanism. Let us move to the third type of mechanism or transport, that is bulk transport. Bulk, as the name suggests, it involves the transport of large number of substances like macromolecules. And it also passes these materials, these bulk material through plasma membrane with the utilization of energy. So that is bulk transport. So these mechanisms, passive, active and bulk transport, they involve in the entry and exit of various materials through plasma membrane. Okay, so this is about the three types: passive transport, active transport and bulk transport. Now after this, we'll talk about cell organelles and their functions. So these are different cell organelles. One is endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes. Lysosomes we generally call them as suicide bags also. Mitochondria, we call mitochondria as powerhouse also, or storage battery of cell. We'll discuss why. Plastids and vacuoles. So we'll discuss all these cell organelles one by one. First, let us talk about endoplasmic reticulum. So when we say endoplasmic reticulum, it is a huge network or extensive network that is, it occupies most of the cytoplasm and found in all eukaryotic cells. When we talk about endoplasmic reticulum, we observe that it is of two types. One is RER, which we call as rough endoplasmic reticulum, and other is SER, which we call as smooth endoplasmic reticulum. Now what is the difference? When we check this RER under microscope, we observe that some grain-like ribosomes are present on their surface. That means some rough surface is observed for RER. This RER is involved in the synthesis and transport of protein. Whereas when we talk about smooth endoplasmic reticulum, it is free of ribosomes and it is present near the peripheral part of cytoplasm. Basically, the products do not pass into the lumen of endoplasmic reticulum in the case of SER, and it is free from ribosomes. The function of endoplasmic reticulum basically is support, transport of material, exchange of materials. All these are the important functions. So support to skeletal framework of cell, transport of materials, exchange of materials between cytoplasm and nucleus, sometimes detoxification. Detoxification is removal of the poisonous substances. Basically, it takes place in liver. It removes the harmful materials from the liver by excretion, and sometimes it helps in the formation of organelles also. So these are the important functions of endoplasmic reticulum. So these are some important functions of endoplasmic reticulum. Now we'll move to the ribosomes. As we have studied that ribosome is present on endoplasmic reticulum to make it RER, rough endoplasmic reticulum. They are basically dense, spherical and granule-like particles. And when we talk about ribosomes, we say that they are protein factories because they are responsible for the synthesis of protein in the cell, and thus we call them as protein factory. So they provide space for the synthesis of protein. That is the important function of ribosomes. Let us move further and we'll talk about Golgi apparatus. The next cell organelle we have is Golgi apparatus. So what is Golgi apparatus? It is sometimes also known as Golgi complex, as it was first discovered by Camillo Golgi. It is present in almost all eukaryotic cells with an exception of red blood cells and sperms of mammals, in which they are not present. The function of Golgi apparatus is packaging of material, synthesis of various cell organelles, and production of yolk in eggs is also one of the important functions for Golgi apparatus. Now let us move to the next cell organelle, lysosomes. We generally call lysosomes as the societal bags. So it has two important functions. One is the digestion of useful materials, and we call it as intracellular digestion, because in this case the organic substance or the material to be digested is taken up by the cell in vacuoles from the environment, and then they are getting digested by the lysosomes. So we call it as intracellular digestion, which takes place in the cell only. Another function is digestion of harmful materials. There are some harmful materials or foreign particles which generally our body don't take. So the important function of lysosome is they digest such harmful materials in nature to defense. So this is a very common phenomenon, natural defense, to save our body from the foreign bodies or foreign particles which may be harmful for our body. Let us move to mitochondria. So mitochondria, known as powerhouse or storage site of cell also. Why this is so? Because they are the main site of cell respiration, and another function is they are capable of self-duplication, which we generally call as replication. So mitochondria we can easily see under light microscope also in the form of very small spheres. They are found in almost all eukaryotic organisms. So the important function is main function is cell respiration. Next we have is plastid. Plastid is, they are the cytoplasmic reticulum. Again we can see through the microscope. And when we talk about plastid, first thing is sometimes they are responsible for the color also. Like we are having different types. One is the leucoplasts, which are colorless plastid, and another is chromoplasts, which are colored plastids. So when we talk about plastid, they help in photosynthesis, as because of chromoplast they have green chlorophylls and they impart color to flower. That is again very important, because once the flowers are colored, they can easily attract insect for pollination, and they store food. So these are important function for plastids. Let us move to the next point, that is vacuole. Vacuole is another cell organelle, and they are the storage bags of cell. When we talk about vacuole, we generally say that they are storage bags of cell because they store material. It may be solid, it may be liquid, it may be sometimes the toxic material also. So they are the storage bags. So they maintain water balance. These two are important functions of vacuoles. Now at the end, let us discuss the difference between plant cell and animal cell. So animal cell small in size, whereas plant cell bigger in size. In animal cell, cell wall is absent, in plant cell, cell wall is present. In animal cell, plastids are absent, cell wall plastids are present. Animal cell is enclosed by plasma membrane, whereas plant cell enclosed by plasma membrane as well as cell wall. So double protection is there for plant cells. Animal cells the vacuoles are many but they are small sized, but in plant cell vacuoles are very few but they are large size. So this all about is the difference between plant cell and animal cell and the complete chapter. Thank you, and if you like this video, please put a like button and subscribe the channel. Thanks.